Hello, everyone, and welcome to today's lecture on stem cell and gene therapies and their impact on biomanufacturing and society. My name is Pedro. I'm a postdoctoral research fellow here in the Department of Biochemical Engineering of UCL, and today I'm going to talk to you a little bit about my research and the wider research within the department in the cell and gene therapy space. So today's talk is outlined in just in front of you. So we're going to start briefly to go over this concept of cell and gene therapies, what they are and what can they be used for. Then we'll move on to touching on some clinical successful examples of when these cells have been applied and how they have transformed healthcare, in particular some forms of cancer. Then we'll move then on our main contribution to the field, which is focused on the manufacturing processes. So we are facing a scenario these days where these therapies have been developed already and deliver in many instances curative outcomes. But the challenge lies on making them affordable so they can save the lives of many more individuals. Then we'll move on with a brief overview over the current projects that the department offers in the cell and gene therapy space. And we will conclude today's talk with a little bit of a teaser regarding why you would want to join our department here at UCL, so the Department of Biochemical Engineering. So moving on with the again the definition of cell and gene therapies and what are they? You can think of cell and gene therapies as a very particular class of pharmaceuticals. However, these differ from conventional pharmaceuticals in many across multiple different aspects. So if you see here on the left, so conventional pharmaceuticals typically address symptoms or a progress of a certain diseases, whereas cell and gene therapies aim to treat the underlying cause of those diseases. So it goes to the root cause of the problem and tries to fix it there. So instead of, again, of interacting with the existing molecules or processes in the body, cell and gene therapy products use genes or cells to try to treat diseases, again, at the core problem, obviously interacting with the local environment as well. So if you think of conventional pharmaceuticals, they typically require ongoing treatment, so multiple doses of the same treatment. In some instances, cell and gene therapies offer one-time treatments or at least long-term benefits where the patient does not have to keep the treatment ongoing repeatedly. Again, another difference between cell and gene therapies and conventional pharmaceuticals lies in the manufacturing process. So when we think about conventional pharmaceuticals, these have been studied to the exhaustion and have very well defined and established processes. For cell and gene therapy products, these are typically less known and less studied processes and typically considerably more complex in nature as compared to the conventional pharmaceuticals. As a consequence of that complexity and that element of personalization, so some of these therapies within the cell and gene therapy are delivered per patient. So one patient will get one batch of the drug or one batch of the treatment. As a consequence of that, the costs are much higher per treatment basis when compared to conventional pharmaceuticals. So now being aware of what cell and gene therapies are, and again, to highlight what they are, these are either genes or cells or proteins or molecules that have been secreted by these cells that aim to treat a particular disease. It is now important to see how have they impacted society already, to really have a feeling of how cell and gene therapies in particular have transformed healthcare as a whole. So before actually we go into that, it is important to define, cell and gene therapies and describe them as this umbrella term that includes not only somatic cell therapies. These are, for example, stem cells. Also, we're talking about gene-edited cell therapies. So here we're typically looking at CAR T cells, so T cells that, for example, have been genetically modified to be able to recognize tumor cells and to be able to kill them in vivo, so in the patient body. And when we talk about cell and gene therapies, we also include pure gene therapies where we're using some form of viral vector that aims to replace, for example, an absent gene, if we're talking about inherited genetic disorders. So when we use the term cell and gene therapies, it is important to know that this is an umbrella term that includes cell therapies, gene-edited cell therapies, and gene therapy products as well, as I just mentioned. So again, a few examples of successful cell therapies in today's day and age. So some of you would have been familiar with perhaps bone marrow transplants. So this is perhaps one of the first cell therapy products that existed, or one of the cell therapy moments that has been described in literature. Typically, patients suffering from some form of an hematological malignancy, so a blood cancer, effectively, typically leukemia. One of the main treatment routes involves chemotherapy and then injecting a stem cell therapy. This is effectively using an hematopoietic stem cell, so a blood-producing cell product whose goal is then reconstitute the patient's immune system. So what happens when a patient has leukemia? Again, this particular line of treatment lies in eliminating the patient's immune system using chemotherapy, and then that same immune system is going to be reconstituted with this stem cell therapy. So this is perhaps the number one, or the first cell therapy application that we know of, but there have been many others in the meantime. This is perhaps the most recent one. So CAR T cell therapies work in a slightly different way, also targeting primarily hematological malignancies, so blood cancers. So the way it works, instead of reconstituting the patient's immune system, instead of doing that, what CAR T cells do is exactly recognize the tumor cells in vivo, so in the patient's body, binding to those tumor cells and then killing them in a specific manner. So the way this product is manufactured, we will see a few slides down, but essentially these, again, CAR T cell therapies have the ability to recognize tumor cells in the patient's body and kill them without killing other cells. So this has been transformational, just for you to have an idea. So CAR T cells, up until recently, and for most of the conditions, were the final line of treatment, which means that these patients have exhausted all the other treatment options. So they were unfortunately not given much more time to live. But thanks to the CAR T cell therapy, they have survived and are here to tell the tale. So CAR T therapies were indeed transformational for these patients. So if we move on to the next class of therapies, so gene therapies, so these are particularly powerful for in the field of rare diseases, especially in those inherited monogenic disorders. And the idea is to deliver that gene that is missing in someone's DNA, to try to stop from whichever problem happens downstream from happening. So you see a few examples here, with probably the most recent one this product being approved called Casgevy. So the first product approved using CRISPR-Cas9 technology that is actually manufactured in Edinburgh at the moment. So we're now going to focus specifically in gene-edited cell therapies. In particular, we'll go over the CAR-T manufacturing and CAR-T, using CAR-T as a case study, because the research that I do here in the Department of Biochemical Engineering is primarily focused on this modality. So since 2017, Professor Kazim Rafik here at UCL has pioneered the CAR-T manufacturing field, and we have seen through the several iterations of projects that we have had within the group, we have demonstrated that we can successfully manufacture CAR-T cell products using stir tank bioreactors. So we were the first ones, or Kazim Rafik was the first one leading this work, and was a true pioneer in this field. So for you guys to have an idea on how CAR-T work, and I touched this a little bit in the previous slides. So imagine that your target cell, and I believe you can see my mouse here, imagine that your target cell is your tumor cell, is the cell that you want to effectively destroy, that you want to kill. So what's going to happen is that the target cell that is in the patient's body, the patient that has been diagnosed with a hematological malignancy, upon administration of the CAR-T cell product, the CAR-T cells are going to bind to the target cell. They are going to become activated, which will then trigger the recruitment of other CAR-T cells to that particular part of the body. So this is going to recruit other CAR-T cells that will therefore start producing these small molecules here called perforin and granzyme that will then be responsible to destroy the target cells, our tumor cells. And it's essentially how the CAR-T cell product works in vivo. Here in front of you, you have the manufacturing process on how all of these works really. So when we're talking about CAR-T cell products, it is important to note and to mention that all the products approved so far and are autologous products. This means that one batch of the drug is going to be administered in one patient, and the starting material to manufacture the product has actually been collected from the patient. So all these products use a similar manufacturing method to the one that you see here in front of you. That essentially starts with leukapheresis, which is this word to describe the process whereby mononuclear cells are removed from the patient, so are isolated from the patient. Then in the lab, so the first step number one, typically happens in a hospital facility or in a leukapheresis center. Then the second step and so forth and so on happens already in the lab, so in the manufacturing facility. So the second step is designed to remove a small subset of blood cells from the whole blood, or from all the nucleated cells. So in this case, because we're talking about a CAR-T cell product, we're interested in T cells. So these are CD3 positive cells that we aim to isolate from the peripheral blood mononuclear cells. Once they have been isolated, and in order to be able to manufacture a meaningful dose to be injected into the patients, we need to activate them. So this activation step is designed to stimulate cell growth. So again, we can reach the therapeutic dose. Upon activation, what we do is to genetically modify these T cells. These can be used a viral vector or non-viral methodologies. In my particular case, my research focused primarily on non-viral methodologies, but we have plenty of successful examples of viral-based genetic modifications here in the department within the work that Professor Kazim Rafik is leading. And then once the genetic modification has taken place, we expand the cells to ensure that we reach the minimum dosage required to administer into the patient. So what does this genetic modification exactly is designed to do? So these T cells, upon genetic modification, we will have in their genome this chimeric antigen receptor transgene that is responsible for the tumor recognition abilities that the final product will have to have. And that's why we need this step. So upon expansion and upon reaching the patient dose required, the product will go through several steps of purification and concentration therefore moves on to storage, and then finally product administration. So here, the work that we conduct in the Department of Biochemical Engineering is focused on optimizing the existing manufacturing processes so we can decrease the cost of those therapies. So many other individuals that unfortunately suffer from these diseases can access to those transformational treatments. So this is our main challenge, to decrease the cost of manufacturing, not only of CAR T cell therapies, but of other cell and gene therapy products as well. So my research in particular, as I mentioned, is focused on non-viral gene delivery mechanisms, and now for the next few minutes I'll touch on these and how can these effectively be delivered in the lab. So if you imagine that we have our cells here, so the way that non-viral gene delivery methods work is that we have to create some sort of simulation to create transient pores on these cells. So you'll see here on the next step that the pores on the membranes have been created. Once this has been established, we then place these cells with nucleic acids. These can be mRNA, these can be plasmid DNA, these can be mini circles. And within these plasmids, there's going to be a sequence that codes for a particular protein and for a particular functionality. In our case, the gene of interest that we're trying to knock in, so to insert on our T cells, is obviously the chimeric antigen receptor transgene. So the way this works again is we start with our cells, we create some form of stimulation to create those transient pores onto the membranes that allow the nucleic acids to enter the cells in a passive manner, and then code for protein translation, really. So this is the way it works. So this is the process. So the process of creating pores onto the membranes and introducing nucleic acid is called transfection. And we have a few different types of stimulations, as I was mentioning, that can be delivered. These can be physical or can be chemically, primarily, and the type of nucleic acid can also vary depending on your goal. It is also very important to highlight here that gene modifications can be delivered with two purposes in mind. One is knock-in, in which you're trying to insert a particular sequence into the cell's genome, but also can be delivered with the idea of knocking out a certain transgene. You can imagine this in light of a particular sequence causing a disease. It may be in our best interest to knock out that sequence to ensure that that defected protein is no longer being expressed because the gene is no longer there. But essentially, this is how non-viral gene delivery methods work. So when we're talking about knock-in and knockout, it is important to touch on the different mechanisms that can deliver these knock-ins and knockouts. Perhaps the most famous one, CRISPR-Cas9, as you may know, but there's others such as zinc finger nucleases and talons as well, and all these three strategies have been primarily used for knockout rather than knock-in. So in all these cases, the way this works is that donor DNA template needs to be provided in order to enable homology-directed repair, where these cells repair the part of the DNA that has been broken down, using this DNA template as a reference, integrating the desired sequence into the genome. So although these three techniques are extremely precise, the knock-in efficiencies are typically low, and it is important to highlight that these have been mostly used for knockout sequences, for knockout purposes, predominantly. So just for you to have an idea of Professor Kazim Rafiq's contribution to the CAR-T space and some of the work that has been conducted here since 2017, 2019, we started by, or Professor Kazim Rafiq led the work that started by demonstrating that we could expand T cells in a stirred tank bioreactor. Then moved on demonstrating that we could manufacture, so expand these CAR-T cells in the stirred tank platform as well. After that, in a collaboration with Sartorius, there was a project to modify one of the existing vessels of the automated platform to Amber 215, and more recently with the work carried out by Tiffany Hood and Pierre Springwell in their PhDs, we demonstrated that we could dramatically improve process efficiencies by establish a perfusion-based feeding strategy. Right now, again, my contribution to the CAR-T work conducted here in the group has been in the non-viral space, with the primary goal being now, can we manufacture a CAR-T cell product using a non-viral workflow? So that's the main goal at the moment, and we're on the verge of being able to publish that work as well. So yeah, these are some of the publications that were generated within the group in the CAR-T cell space. And here are the three main figures that lead the cell and gene therapy activity here within the Department of Biochemical Engineering. So we have Professor Kazim Rafiq, so professor in bioprocessing and biomanufacturing of cell and gene therapies. So Professor Kazim Rafiq is a world-renowned specialist in large-scale manufacturing, initially of stem cells and mesenchymal stromal cells in particular, but now as professor in CAR-T cell therapies and in the viral vector space. We also have Dr. Rana Khalife, specialist in induced pluripotent stem cell research and leading our stem cell and gene therapy manufacturing and commercialization master's program. And recently we were also joined by Dr. Bernice Wright, who's a specialist in, or has specialized her research interests around using stem cell secretome and extracellular vesicles as well. So these three key figures are responsible for the vast majority of the thought models within the cell and gene therapy space. So if you do join us to study, whether it's an undergraduate level or master's level or even PhD level, you'll certainly meet them, and we'll see these three key figures in some of your lectures. So without anything else to tell you, I'll finish my talk right now, and I'm happy to take any questions from now.
Fantastic, thank you very much. Oh, I've just had a little thing come up saying that my internet connection isn't stable. Can you hear me okay?
We can hear you okay, yes.
Okay, right. If I do drop out, then it's because let's just say BT's in for some trouble. Anyway, we seem to be okay at the moment. So just to let you, whilst the questions come in, please put your questions in the Q&A box. You can post them anonymously. Some of them Rana has answered in text, some of mine answered in text. It's more if it's easier just to send you a link to something where it's appropriate. But please put your questions. I can see they're coming through thick and fast. But what I'm going to do is I'm also going to put a link to the UCL Undergraduate Open Day, We've got 2 sessions where myself and Dr. Jack Jeffries will be talking to you about our undergraduate degrees here at UCL Biochemical Engineering. So you can talk to us about content, careers, any aspect of studying degree. If following this session you're interested in doing an MSc in manufacturing commercialisation of stem cell and gene therapies, both Bernice and Rana will be available on the 15th for a Q&A session. So you can ask them anything about the programme, about what the career, but now they've got a cohort of people who've actually graduated and have gone on to careers. So you can talk to them about the amazing opportunities. You can also find out about the bursaries that we've got available. So we've got 2 £7,000 bursaries for this programme, which is really great to be able to offer that. So if you want to find out more about that, please join us, or just click on the prospectus entry. But both Rana and Bernice will be available. I'll put links to those in the chat. But now that I can see an absolute list of questions have come through, I'm going to hand over to Bernice, Rana and Pedro. If you'd like to just, between you, suggest who wants to answer it, answer the questions, go through them. I'm really sorry, we won't have time. We're unlikely to have time to get through all the questions, but we'll do our best, okay? So bear with us. And I'm going to put some links to events coming up so you can find out more about degrees and postgraduate degrees. Over to you.
We haven't introduced ourselves. So my name is Rana Khalife. So I've got a lot of questions regarding BEng, MEng, and BSc of Business and Management. So I'm the chair of the recruitment in the department, and I'm the regenerative medicine minor coordinator. So if you're doing a BEng, MEng degree within the Faculty of Engineering, usually the faculty will offer 22 or I don't know, this year 18 to 20 different minor. So I'm leading on the regenerative medicine minor, and I'm the MSc program director of one of the MSc that we have, which is manufacture and commercialization of stem cell and gene therapy. So this is what Kim was asking you, like if you're interested in this MSc, you will have a Q&A on the 15th, I guess. So feel free to join us. My background is I'm a biochemist, but I did my PhD in biochemical engineering. And what I could tell you that my perspective of seeing things have changed dramatically with biochemical engineering. I'm not being biased, but this is the truth, because it's basically we are trying to translate research. So Pedro presented the research and what's going on in industry. And because we are heavily linked to industry, we try to optimize and teach the students whatever is happening in industry. So once they graduate, they can see how real life is. I've got a lot of questions regarding after finishing your degree, what's the difference between BSCs and BEng, MEng. Now, BSc will focus on the business side along with the bioprocessing aspect, while BEng, MEng, as I mentioned, you will take a minor, but you will focus more on the engineering aspect, design project as well. Now, it's really difficult to say that all of the students will get this kind of a job after they graduate, because they have a really variety of options once they graduate. So we have people who are process developers, scientists. We have people working as a consultant even, and on the business and project management. So you will have the chance to have different kind of variety of jobs. Even some of the students finished, continue with us for their MSc and PhD as well. So I'm biased for stem cells, a bit of CAR-T as well. So you can ask me a lot of questions on stem cells. I don't know, Bernice, do you want to introduce yourself and then we can go over the questions?
Thank you, Rana. Can everyone hear me? Great. So I'm a teaching lecturer on the MSc in commercialization and manufacture of stem cell and gene therapies. I work with Rana and with Pedro and with Kasim as well. And I teach some of the main modules on the MSc course. So I would teach you about the pharmacology, the commercialization, and the development of ATMPs, advanced therapeutic medicinal products, that includes the stem cell therapies and CAR-T therapies, and also gene therapies. and some of the more the newer therapies like extracellular vesicle therapies, so cell secretomes, as Pedro mentioned, and this is my research interest. And I also help, I mentor many of the students, and I help students find graduate positions after they finish the MSc. And as Rana mentioned, quite a few of our students go into industry. Some of our students decide that they want to continue research, because one of your main modules in the summer will be a research project, and this is the most heavily weighted module on the MSc, and it's quite a lot of fun. You get to work in the lab, you get to work with industry. Many of our students are interested in working in industrial labs, and because one of the unique features of this MSc is working very closely with many of the UCL spin-out companies. So we have very close links to industry. You have a good chance of working during the summer on your research projects in a company. So if anyone is interested in taking that path, they have this as well. And we have many very exciting ND and PhD opportunities available in collaboration with some of the major biopharmaceutical companies in the world. So AstraZeneca, Roche, Paul, some really great collaborations. So, and two of our current cohort have now got NDs working in the department. And so you can go either path, the academic path or working with industry. So I can take any of your questions on the modules that I lead, and also generally on the MSc course, and working, if you decide you want to go into academia, you want to do a PhD, I can answer questions about that as well.
Yeah, so let's just make it clear that what we're talking about as a MSc is the MSc Commercialization and Manufacturing.
Yes.
Some of the attendees have asked me, is there always an exam? So on our MSc Commercialization and Manufacturing, there is no exam, so it's going to be only coursework. But with the other MSc program in our department, yes, there will be exams. And as Bernice have mentioned, the unique thing about this program is that we have secured kind of like, not internship, but kind of like research project based in industry for our students for the second year. Now, for the other MSc program, is there any internship opportunities? It depends. So whenever we have the chance that our spin-out company or the company that we collaborate with send us that we need students, we usually advertise it to our students. But this is not always, like, it depends on the company.
Mm-hmm. We do have one student who's working with Orchard Therapeutics. He was very successful in getting an internship at the beginning of the MSc for 2 days, I think, a week, or 1.5 days a week, working as an intern. So it's possible to get a part-time internship with one of the companies that is affiliated with us, or just another company, which is a cell and gene therapy company. If you're proactive enough and you go out there and you make that link with them.
So what are some path into research and work relating to stem cell therapy we can take after completing an undergraduate degree? So as I mentioned, even with the stem cells, because there are a lot of spin-out companies from UCL, from the department, and they're all startup near London as well. So you can be working in stem cell industry. So again, it could be related to Q&A, quality control, quality attribute, or bioprocessing developer, or like a scientist. Even you can be doing something as a desk-based, just like kind of a business plan or project manager as well, management as well.
I see a question for perhaps Pedro could answer. Can CAR T cell therapy be combined with targeted inhibitor therapies for enhanced treatment outcomes for leukemia? It's very specific.
It is a very specific. Could you repeat?
Can CAR T cell therapy be combined with targeted inhibitor therapies for enhanced treatment outcomes for leukemia?
Yeah, definitely. So there's some clinical trials exploring CAR T cell activity not just for leukemia applications, but also for solid tumors, and not just on their own, in combinations with multiple other therapeutic approaches, even chemotherapy together. So it's not exclusively just CAR T on its own. So it can be used as a combination therapy as well. Does that answer the question? I hope so.
There's
Go ahead, Bernice.
there was a question on gene. When faulty genes or parts of genes are removed, are they replaced by useful genes during the treatment? Yes, this is what the basis for gene therapy. So either a gene can be repaired or it can be replaced by gene therapy. Or gene editing therapy. I'm sure everyone is familiar with the latest gene editing therapy, Casgevy, manufactured by and developed by Vertex and CRISPR Therapeutics. So yeah, this is the basis for these types of therapies. I don't know if anyone wants to pick up. Is there a future for gene therapies in
I was going to
xenotransplantation?
answer this, but go ahead if you want to answer. I
Um,
mean, so for xenotransplantation, okay, so first you have to check where are you based, which are kind of regulatory body that you are following, and to see how you can proceed with these things, because these are derived from animal. Okay, now usually with tissue engineering, especially like for the trachea one, because the trachea shape is hard to mimic, usually we take them from pigs. So from animal, we decellularize, that means we remove all of the animal components, and then we put the cells from the patient, and then we grow them together and then give it to the patient. Then we can use these kind of products that are derived from animal. But I'm not sure about the question, how could a gene therapy be used in xenotransplantation? Because it's really different. So if you could clarify for us, that would be great.
There is a very nice question about affordability of gene therapies. Of course, we know that the price of gene therapies currently are quite prohibitive. And I think we can all comment on that. Currently, we are trying to reduce the cost of manufacturing. So some of the components of a gene therapy, for example, a viral vector, they're very expensive to manufacture. And if we drive down the cost of manufacturing, we can bring down the cost of these types of therapies. And this is currently one of the bottlenecks that we're trying to address with the research that we do.
Yeah, it's one of the main things that we teach our students is, okay, so you have the research, you have the innovation. Now, our aim in the department is how you translate this into real life, because it will take time. Not only translate it into real life, how you can produce this at a larger scale. And the most important thing is how you can reduce the cost of manufacturing to make it affordable for everyone. That's why we collaborate with a lot of companies, that they will be like, okay, we're looking into this bioprocessing. Can you optimize this for us so we can lower the cost of it? There's still a lot of debate. So with the first CAR T, Kymriah, it's 475,000, and then when they tried to sell it to the NHS here, they were trying to negotiate the price, and then they gave a special price for the NHS as well. But you have to think about it that this is a one-go treatment, whereas if the patient is having sort of like always another treatment, so what will be the cost of both treatments in comparison to each other?
There's a very nice question about gene editing therapy. So do we just modify DNA in one single cell, or are they modified in batches? So currently, gene editing therapy, for example, we know about CRISPR, the efficiency of these types of therapies are quite low, and the cells are modified ex vivo. So the cells are taken out of the body, they're modified with the gene editing therapy, and it's not just a single cell, it's quite a number of cells that need to be modified and then reintroduced back into the body as the therapy. So it's not the most efficient way of developing a therapy, and we are working on that. There are other types of gene editing approaches, prime editing and base editing, and these are they're more efficient, or a little safer than the CRISPR, because there are the problems of introducing mutations using CRISPR, and there are ways of changing the Cas enzymes involved in this machinery so that we can reduce these types of errors. So despite the fact that we have approved gene editing therapy, we still have a long way to go before they become mainstream types of ATMPs, advanced therapies.
Perhaps is also, I think, always important to highlight that so all of these manufacturing bottlenecks that we try to solve here on a daily basis in the department are sometimes brought to us by companies. So the department is very connected, very well connected with multiple companies within the space in such a way that some of our PhD projects, but also master's projects, were designed to solve bottlenecks of our industry partners. So they were designed with the purpose of bringing technical solutions to real-life challenges. These are not just academic exercises. So in the Department of Biochemical Engineering, so where we see is a very much applied department. So perhaps less focused on blue sky research and more on solving challenges that the industry faces. So it's really, really close to applications and really, really close to what the industry needs now. So that's why we really feel we're making a tremendous impact on trying to, again, solving some of these technical bottlenecks to reducing the cost of manufacturing. And obviously, as well, if you do decide can join us, whether you're an undergraduate or master's or PhD student, you will for sure have the opportunity to see what our approach and the type of skill sets our programs have to give you the ability to tackle some of these challenges, not only from a experimental design perspective, but also from a technical and applicable perspective.
There's one question. We have other MSCs available in the Manufacturing Futures Labs at UCL East that do address sustainability. So this is a question. So one of them is Chemical Sustainability MSc. We have another very nice MSc, the one in Nature Inspired Solutions, and our advanced materials, so digital manufacturing of advanced materials that can also be linked to sustainability as well, in the way that we want to produce more environmentally friendly solutions for some of the problems that we have with manufacturing fuels, with agriculture, for example, new agricultural approaches. So yeah, we do address sustainability in the MSCs that we offer at UCL.
Yeah, I mean, I think part of the question is as well within our MSc, which is commercialization, what is the scope of the project? So it really varies. So last year we had sustainability, 2 projects actually on sustainability, one within the UCL hub and then another one with industrial partner and consulting. So we have a really variety of projects. We have CAR-T, we have Lenti, we have stem cells, we have desk-based, we have business plan, and then we have the industrial project as well.
Mm-hmm. So there's a good question about CRISPR. How can CRISPR be used to make gene editing more efficient? Well, we can have a multiplexing approach with CRISPR that can make gene editing more efficient. And without going into the technical details for this, I mean, you can look into this. I can put into the chat some very nice reviews, or there are nice reviews available that you can find, even if I can't find them immediately right now, about the types of approaches that you can take, where you can use different types of Cas enzymes to multiplex CRISPR, and that will make it more efficient. So we are working on approaches to do that. There is a question on, perhaps Rana could pick up on stem cell therapy for antimicrobial resistance.
I'm not sure which kind of disease, like antimicrobial resistance, but which kind of stem cell. So if you could elaborate more, that would be good.
Just whilst we're waiting for somebody to, if you could expand a little bit on more your question about antimicrobial resistance and that, I just want to remind everybody that if you want to talk in detail about the program, both Rana and Bernice will be available on the 15th. There is a link in the chat. If you didn't get a chance to open it or save it, if you simply go to the front page of our website, it will be available there. Also, if you'd like to know more, and I know that a couple of people were asking about the degrees here in biochemical engineering. Myself and the admissions tutor, Dr. Jack Jeffries, will be available online later this month. You can come and talk to us, and we'll tell you. We'll have someone from UCL Careers as well who can talk to you about hopefully talk to you about careers. But the best thing of all, if you could possibly do this, would be if you could come and meet us in actual person and see our actual amazing labs and meet our actual amazing people. They do really exist, but seriously, our laboratories are unique in this country. Nobody else has anything like them. We have a pilot plant where we can actually pilot processes, which is an incredible facility to have, and we'd love to show you around. So that's happening on the 28th and 29th of June. If you register your interest, you'll be first on the list to get invited along to that, but pop it in your diaries, save the date. If you can make it to London, and it's also a chance to see the campus, see the amazing buildings that we have, and I think you'll even get to go across to UCL East, which is where Bernice mainly works, which is a whole new campus we've got. Somebody's just asked whether we're visiting the summer school in India. We don't go from our department because there are so few of us. We don't have the chance to go out, and unfortunately we don't get to go out and visit people in India. The furthest any of us has got is I got to go to Hull once to meet people, so that's as far as we get to go. I'm afraid not, but there may be someone from UCL coming along. But if you can come here or come along to the webinar that we're doing, I sent the link for that. So I think we've come to the end now. Yes, somebody's just expanded on their question. So the last question that we've got will be for, somebody said that they're going to be in for the Offer Holders Day. That's great. Yes, you'll be meeting me at the Offer Holders event. Yes, so just the last question there for you, Rana. Somebody's expanded on their question. I can't even pronounce that.
We do work on I work on mesenchymal stem cells right now. I can offer, and I mean that's an interesting idea to work on this for methicillin resistant Staphylococcus aureus MRSA, and this is not something that I know anybody who's doing any research on this antibiotic resistance in the department in this aspect, but it's certainly interesting, and I mean, I know, so I'm trying to think back on research I did in the past, but no, this is not something I've worked on. We do, yeah, I mean, it might be an interesting project to work on.
Exactly, I was going to say, why do you come spearhead it? You know, this is one of the things that really marks UCL out as being is a difference that we have, is that our students have access to our facilities, they participate in our research programs, they're really fully integrated. It's a research and teaching environment. So somebody's just asking about the labs. That's on the 28th and 29th of June. We'll be doing tours, so they'll be with our postgrad students as well, so you get to speak to people who are doing their research there as well. Okay, we have come to the end of this session. I think we've yes, there will be detail. What I'll do is I'll send a link to all the things that I mentioned. So the offer holder day, the open days. I'll send a link to that when I email people and let them know about this event. So and I'll also send you a link to the recordings of the other events. So if you didn't get to see the one that's coming up on autoimmunity with Dr. Thomas McDonald, then you'll get to see that one as well. All the others that we're doing. So I'll send a link to that through. They'll all be available on YouTube. But finally, I just want to thank you ever so much for joining us. I hope you really enjoyed it. It was great to have you with us. Thank you, Pedro, so much for taking the time to participate in this. Thank you, Bernice. Thank you, Rana. But most of all, thank you all for joining us. Really appreciate and your kind words. Yes, we really thank you as well. But it's great to have you with us, and we hope to see you in person sometime in UCL.
Bye.